It's my pleasure to introduce our speaker, and I feel like now talking to science gives you a lot of pressure, right? And Dr. Richard Watanabe, and who started as a postdoc with Mike. You're Mike's first postdoc in the year of 1995 to 1998, and Richard started doing type 2 diabetes genetics research, both method development, and back then probably like earlier days of fusion. I think probably back in those days you were still calling like the NIDDM, like the diabetes mellitus, instead of type 2 diabetes. So Richard actually has still, like working on, genetics of type 2 diabetes or diabetes in general, and also related glycemic and other traits since then. So. Richard will talk, is currently a professor in the Department of Population and Public Health Sciences, also the Associate Dean of the Health and Public Science Programs at University of Southern California. So welcome, Richard.
Thank you, Yoon. It is really a tremendous pleasure to be here and to be talking to all of you. As Yoon pointed out, I started with Mike way back in 1995. And it was really, for at least me, I tell students all the time when they ask about career advice and things like that, my career has been a series of dumb luck, right? I met Mike for the first time when it was either the second or the third planning meeting for the fusion study was held, and it happened to be in Los Angeles, and my PhD mentor, Rich Bergman, was hosting the meeting in our lab, and Rich comes down to my office and says, we're having this meeting about genetics. I think you should sit in. And I'm like, why? Because I don't do genetics. I did read some paper by this guy named Philippe Fragel, who had just discovered genetic variants for glucokinase for MODY2, and that's about my extent of knowledge about diabetes genetics at the time. But in this room was Mike, and this other guy named Francis Collins, this other guy named Jaakko Tuomiletto, who actually I knew Jaakko, and then there's this other guy named Sumitra Ghosh. And I didn't understand a single thing that was being discussed. I understood the word chromosome, genetics, and gene, because as the title says, I'm a physiologist by training. But that was about the time I was looking for postdocs, and at that time, physiology was appearing to be a dying field. And so I decided to seek guidance from my boss, and Rich was like, you should really do genetics. And so I reached out to Mike, and from my perspective, Mike took a risk by letting me do a postdoc with him because, again, my knowledge of genetics were those three words. And yet we were, he was starting off on this project to, find genes for diabetes. And it's turned out to be a lifelong positive thing for me. I enjoyed my time here at Michigan, but even after I left, I could always pick up the phone or I could send Mike an email to get advice and chat and whatever. And he's been a wonderful mentor and friend. And really, my time here at Michigan with him and his group really enriched my career, to extents that I can't really, even today, fully understand. And I can't thank you enough for everything. So since many of you in the room, this is really sad too, many of you weren't born in 1995. And for legal reasons, I have to put this slide up. So anyways, so I just thought I'd review, right? So what did the world look like in 1995? So the world population, a little over 5.5 billion people. The US population was only 266 million. Our president was Bill Clinton. Average life expectancy was only 75 years, right? So we've gotten a lot better. That's gone up. And you can see every one of these teams is not doing very well these days, except maybe the Braves. But like the Rockets died just recently in the playoffs. The New York Islanders have never recovered. And then there were other world events that occurred. This is only a partial list. I had to point this one out, that Mississippi was finally ratified or approved the 13th Amendment. It only took them 130 years to do it. We had our first sort of major domestic terrorist event, the Oklahoma City bombing. Windows 95, yes, there were other versions of Windows before the ones you're currently using. And I still don't understand the numbering system that goes from Windows 95 now to Windows 11. It was introduced by Microsoft. It was the 50th anniversary of the atomic bombings of Hiroshima and Nagasaki. PlayStation was released, right, way back when. Toy Story, the first full-length feature film that was entirely generated by computer animation, which I'm sure ChatGPT today could spit out very quickly. The first protease inhibitor to treat HIV/AIDS, sequinavir, was approved. Sadly, the last Calvin and Hobbes comic was published. I miss Calvin and Hobbes. And I've saved this for last, O.J. Simpson found not guilty. Again, some of you were around when the O.J. trial was going on. A lot of you weren't. If you weren't, there's a TV series, apparently, that miniseries that goes over it. But I saved that one for last because that was the first time the public really got to know genetics, because the entire trial was televised. And they had all this DNA evidence, and there were, the experts testifying about this stuff, et cetera. And people learned that you can actually use genetics as forensic evidence to try, convicted potential criminals, et cetera. And it basically introduced this thing you now all see on TV, where in every sort of cop show, right, somebody picks up a little piece of something, and suddenly the next day, all the DNA evidence is available to you, right? But the other thing was, it was not too long after I got to Michigan that we had Bruce Weir visit. And Bruce was actually an expert witness in the O.J. Simpson case. And for those of you who don't know what happened, I'm not going to say anything. You could go look it up. But Mike, we had a lab meeting, and Mike was very explicit. No questions about the O.J. trial, right? And then the Nobel Prize for Physiology and Medicine, I just wanted to point out, that was awarded to Lewis, Nussland, Volhard, and Weichenhaus, was related to discoveries about genetic control for early embryonic development, right? So this also sort of injected genetics into the public eye even more. When I showed up to Michigan, the fusion study that many of you know had just gotten started. I think we were in like the second year maybe of data collection. So we were going after affected SIB pairs in Finland to do genome-wide linkage analysis. And we were going to find all the genes that way, right? And around that time, right, a year later, the Gene Hunter paper was published by Krugliak, right? So this was the methodology that we were going to use. And as soon as we were deciding to do that, the landmark paper by Neil Risch and Mary Kangas came out that said, screw it, you should be doing association analysis, don't bother with linkage. And all of this wonderful statistical genetic methodology that was developed, everything that we were using as tools, were being implemented on things like the Sun microstation that was running Unix, and of course the early versions of the PC. This is actually a IBM AT that came with one of the best keyboards ever, right? The IBM, the Model M keyboard, which had this very positive tactile feedback. It was nothing like the mushy keyboards that we use today. So when you walked by Mike's office, that's what would be emanating, right? Which just shows you that Mike was busy, working on something. Of course, this was also the time when word processors were first being widely utilized. And I still remember the first Word document I got from Mike that he asked me to look at and edit. I'm sitting there trying to make changes, and I'm noticing that the lines aren't aligning correctly, right? I delete stuff and the line would get shorter. If I added more text, it would just get longer. And it wasn't wrapping around. I was like, this can't be right. And I'm trying to figure out what's going on. And finally, I turned on the paragraph indicators. And Mike had not yet transitioned from using a typewriter to a word processor, because at the end of each line was a carriage return line feed. So this is a picture of a much younger Mike. This is when we had a fusion meeting in Bethesda, Maryland. This was maybe a couple of years after we got started. But here are the 4 PIs. So here's Mike. This is Jaco Tumoletto. This is Richard Bergman, who's my PhD mentor, who was my PhD mentor, I should say. And of course, Francis Collins. And we've come a long way since then. And as I said earlier, this was in a very enriching time for me, because not only did I get to sort of throw in my 2 cents about what I knew about diabetes into this whole process, but I also learned a whole, what load of new things that I incorporated into my own career going forward. So I'm about to give you guys a very disjointed talk because I wanted to hit 2 things, 2 different topics that are kind of related, but come from the perspective of me being a physiologist doing genetics, okay? So one thing to remember is that glucose, right, which is the key thing we use to diagnose diabetes, is it's one of the most highly regulated things in the body. Okay, and there are a whole number of different mechanisms that are used to try to keep glycemia in the normal range. And in the field of diabetes, there's been this long history of trying to identify the primary cause of the disease, and there were large arguments surrounding the idea that it had to be either insulin resistance or insulin secretion. And the reality is that that sort of binary thinking wasn't appropriate, right? So we can't look at insulin sensitivity, insulin secretion in isolation of each other, because there's a regulatory feedback cycle, right? So any change in glucose is going to engender a change in insulin. And so you have to look at both of those things sort of simultaneously, okay? And the idea is that the pancreatic beta cells are really trying to balance these things. How do I secrete enough insulin to regulate glucose production and uptake, right, so that glucose stays in the middle? So pancreatic beta cell function, right, is the pancreas secreting sufficient insulin for a given metabolic demand, is really the question that we should be focusing on. And therefore, we needed a way to try to quantify this activity of the pancreas. And my PhD mentor, Richard Bergman, who unfortunately couldn't be here today, he was planning to be here but had to have hip replacement surgery last week, and therefore can't travel, so he sends his apologies. But he, back in 1981, introduced what was called the disposition index. And this is basically a hyperbola that's defined by insulin sensitivity and insulin secretion, as shown here. The formal disposition index, as it's used today, uses insulin sensitivity derived from what's called the minimal model. This is a mathematical model that analyzes data from intravenous glucose tolerance test, and the acute insulin response. That's what AIR is, which is the first phase insulin secretion. This quantity, the disposition index, is now widely used in diabetes research and in other metabolic research. The problem is you can't simply divide any measure of insulin sensitivity with any measure of insulin secretion, because the nature of that relationship may or may not be hyperbolic. And if you look at the work of Steve Kahn, who got the Banting Award last year from the ADA, he has a wonderful series of studies that actually shows that not all of these relationships follow a hyperbola. Okay. So let's go back and look at that original data. So this is the data. This is the characteristics of the participants. There were 18 participants in that paper. Again, those of you who were not born in 1995, BMI did not exist. So we were categorizing people by percentage ideal body weight. Metropolitan Life Insurance Company would publish these tables with the percentage ideal body weights, and we use that to classify people. So you can see we've got some lean individuals, some obese individuals, and they're either glucose tolerant or glucose intolerant, determined using something called the kg. You don't need to know what that is because nobody uses it anymore. And you can see here, here's the fasting glucose, and you can see the fasting glucose is moderately different across these groups. And then the fasting insulin, we see this general pattern of increase, although going from the normal tolerance to the intolerant people, we see this decrease in fasting insulin. If we directly measure insulin sensitivity and insulin secretion, this is what we get. Okay, so those groups, basically the insulin sensitivity is pretty similar, except in the obese intolerant group. And then insulin secretion, that pattern looks like this. Okay? When you calculate the disposition index, you get what looks like a similar pattern as insulin secretion. But if you look at the percentage differences relative to the lean tolerant group, you can see that insulin sensitivity, again, for the lean intolerant and obese tolerant are kind of similar to the normal, the lean tolerance. Obese tolerance, there's a significant drop in insulin sensitivity. Insulin secretion, we see a drop. Again, there's that drop with the lean intolerant group, and then increases for the other groups. But if we look at disposition index, we see a pattern that's very different from what we're seeing with insulin secretion. And again, that's because the disposition index is taking into account that relationship between sensitivity and secretion. So this is the figure from the page. Fortunately, Rich had a table with all the numbers in it, so I could recreate the figure. And this line, this hyperbola here, is the average across all the individuals. And you can see that the disposition index seems to segregate people with good tolerance versus people with poor glucose tolerance. And this became sort of the thing that Rich pushed. We need to look at beta cell function, and therefore we have a way to quantify that using this method. Now, unfortunately, this is, again, the way the data was presented in the original paper, and ever since then, everybody presents the data in the same way. I do my calculation, I get a number, I plot it on the graph. But remember, this value actually represents a hyperbola. Each individual has his or her own hyperbola. So in reality, this figure should probably have been drawn this way, okay? Because over short periods of time, if your insulin sensitivity, if your metabolic state doesn't change dramatically, then you should move up and down your own personal hyperbola. So one day your insulin sensitivity might be lower, it might be higher, but then your pancreas should adjust the secretion, right, so that you stay on this line. Okay. Now, your hyperbola could change. Okay. You're on your way towards diabetes, no bueno, right? Your pancreatic beta cell function might be dying. So there are two important characteristics of the disposition index. One is the current capacity, your ability to compensate for metabolic changes on the short term. But then there's the longer term where your hyperbola might shift. And so you might have diminished capacity or improved capacity. And we've shown this in a number of different, both in animals and in humans, by inducing, we can't really tweak insulin secretion very well, but we can induce changes in insulin sensitivity using things like nicotinic acid or other drugs. And we can actually see people move up and down. But we can also compare groups with different metabolic states and show that their hyperbolas are in different places relative to somebody that we would, quote unquote, call normal. Okay. So what we really should do, and I'm sorry my multiplication sign didn't come out there, but we should really think about DI and all its components in terms of time. Right? Short periods of time versus longer periods of time. And you can think of it more in terms of what's the change in disposition index. And you can see here again, it's going to be dependent on whether your insulin sensitivity or your insulin secretion appropriately changes under certain conditions. So the key thing here is you can't treat the disposition index as kind of your typical cross-sectional measurement, right? So this is, and in fact, just recently in the last year, I've seen like 3 papers, both of them, 2 that are genetics and 1 that are non-genetic, where people have done this, where they get their data, they plot it, and then they do something, well, let's look at like insulin-resistant people versus not, and et cetera, and they might use something like the median and create these things and say, okay, I've got people who have high sensitivity and high secretion, high sensitivity, low secretion, et cetera, et cetera. And they stratify their data that way, and then they go on and do some other analyses, thinking that this person is there and that's it. Okay? But again, like I showed you previously, it's a hyperbola, right? So again, these people are moving up and down these hyperbola. So if you apply that same sort of stratification, some of these people are cutting across 3 different quadrants of your stratification, right? Depending on when you measured them. But that doesn't mean that over here they have poor insulin secretion, over here they have better insulin secretion. They're just adjusting. And the question really is are they moving? Is their disposition index moving or not? Okay? And the disposition index does move. So here's some example. I just took a bunch of archival data that we had. This represents something like 2,500 or so individuals that we've done this analysis on, and looking at 3 different racial ethnic groups. So if we start with non-Hispanic whites, African Americans, and Hispanics, all with normal glucose tolerance, you can see this is a 3-dimensional representation. So I have insulin sensitivity, insulin secretion, and then the disposition index. So you see these 3 groups, even though they all have normal glucose tolerance, basically they start in different places. Okay? But if I look then at other groups, so if I look at people who have impaired glucose tolerance, if we look at the non-Hispanic whites, you can see their disposition index drops quite substantially. Even though their insulin sensitivity hasn't changed much, they've seen a drop in some of their insulin secretion. And if they go then on to, if we look at type 2 diabetes, we can see that transition. So again, that disposition index is moving towards the origin. Okay? And the same is true for the other 2 ethnic groups. But you can also see here that there's a difference in the general starting points and how they get to type 2 diabetes depending on their racial ethnic group. And that's cross-sectional data, but we can show the same thing using longitudinal data. So this is data that comes from my good friend and colleague, Tom Buchanan. And basically, they did a follow-up study of Mexican-American women with a previous diagnosis of gestational diabetes. These are women who are at high risk for developing type 2 diabetes in the future. And he basically did a longitudinal follow-up of these women for 135 months. So a little over 10 years, right? And he identified those women who remained diabetes-free versus those who developed diabetes. And at the beginning, you can see here, this is the average insulin sensitivity, secretion, and the disposition index, individually, and then on the 3-dimensional graph. And you can already see here that there's a difference between the groups that developed diabetes versus those that didn't develop diabetes. And as you follow them over time, they end up here, right? So again, the women who didn't develop diabetes, the women who did. Okay? And you can, again, see their summary data over here on the right. Okay, so everyone changed. Their insulin sensitivities changed, their secretions changed, et cetera. How do they get from here to here? Well, the women who remained diabetes-free, that's what they did. Okay? And basically what's happening is they're obviously changing insulin sensitivity a little bit. Their insulin secretion is changing in comparison. And their disposition index, although it is dropping at a relatively low rate. Whereas the women who develop diabetes basically are doing a nosedive towards moving towards the origin. Mathematically, that's where we should end up. I don't know if there's ever a case where that's happened. But. And there's more data that out there that basically shows you the same general pattern. Okay? So again, when using the disposition index, we have to be careful on how we treat it. For all you young people out there who are just starting out and maybe doing diabetes research, keep this in mind, because I'd hate for you to do this really fancy schmancy analysis using all this wonderful technologies out there and then make the wrong interpretation because you treat disposition index as a cross-sectional, your typical cross-sectional measurement. Okay, so like I said, this was going to be disjointed. So shifting gears totally. The other topic I wanted to hit on is the utility of non-clinical phenotypes. This is something that drives a lot of our research. We grab data that we readily have available to us, right? Whether it's via medical record review or whatever. And clinical phenotypes are important. I'm not downplaying them at all. But you have to remember, they're clinical phenotypes. They were originally selected to be measured because they had certain clinical relevance. They may not be the best things in terms of mechanistic or other relevance, right? And I think we always have to keep that in mind. So one of the things that we meaning my group, and even Fusion did for a little bit, is we've done additional phenotyping. So my studies, we always do the intravenous glucose tolerance test with minimal model analysis, so we get measures of insulin sensitivity, secretion, we can get the disposition index. We also get something called glucose effectiveness, which I'll talk about in just 1 second relative to this figure. And we also do body composition by DEXA scan. We do oral glucose tolerance tests, which sounds like a standard clinical test, except we do blood sampling every 30 minutes. So that we get a profile that we can use and leverage that information. So the great thing is I have all kinds of great information. The bad thing is it takes time and money to do all this, so my sample sizes are on the small side, right? So I don't have a million cases and a million controls to work with. But we make our little contributions because we learn little things. So one of the things that I was playing with, I thought of was something that I'm calling the substrate balance hypothesis. And when looking through the signals from all the GWASs we've done, I was struck by the fact that there were 3 loci that were kind of primarily expressed in the liver. So glucokinase regulator, and patatin-like phospholipase 3. And last year we published this paper which sort of tried to dissect this idea that, well, GCK and GCKR are related to glucose uptake by the liver, and PNPLA3 is related to metabolism of lipids, basically forming triglycerides from stored fat. And this idea that there's basically a set of substrates coming in, a set of substrates leaving the liver, and that balance between going in and out might be determining how much liver fat is accumulating over time. Okay? And so I'm not going to go through the whole paper. I wanted to focus on taking advantage of one of these phenotypes, and this is that phenotype I was telling you about earlier, glucose effectiveness. So what glucose effectiveness is, it quantifies the effect of glucose to enhance its own uptake at fasting insulin levels. It's basically quantifying the mass action effect of glucose. And based on studies that we've done when I was a student, we know that glucose effectiveness partly, not fully, but partly reflects glucose uptake by the liver. We've estimated it to be somewhere between 25 and 35%. Okay. So my thought was, well, if that's the case, and if these variants actually regulate glucose uptake by the liver, we should be able to find it or see it in this glucose effectiveness measure, and in fact we do. So this is looking at GCKR, right, RS780094, and glucose effectiveness, and you can see that with each copy of the A allele, we see an increase in that uptake, which fits fine with the sort of biological picture. But then it got me thinking a little bit more, because if you think about the fact, again, my hypothesis of we're regulating glucose uptake by the liver, and we're also regulating triglyceride formation through liver fat, right? So GCKR, which encodes for GKRP, this is the thing I hate about genetics, is sometimes the gene name doesn't actually align with the actual protein name, et cetera. For years, I grew up on glucose transporters being called GLUT1, GLUT2, GLUT3 and 4, and then somebody in the genetics field changed it all to some sodium, I forget, I can't even remember the name anymore because it's way too long. But anyways, this mechanism was very well known, and we know that glucose being taken up by the liver goes down glycolysis and goes through the TCA cycle and can feed fatty acids and feed the liver. But there's also the effect of dietary fructose, right? The evildoer that's causing all of our metabolic problems. And fructose, remember, we don't produce fructose endogenously. It all comes from exogenous sources. Yet we have a mechanism to metabolize fructose, and that feeds into, you can see the different pathways and how they feed into the formation of liver fat. But in vitro, there had always been this evidence that fructose 1-phosphate could be regulating GKRP. And there's lots of beautiful in vitro data that shows this. But to my knowledge, nobody had ever shown this in vivo. And my thought was, well, we have dietary fructose information on everybody. We have this unique measure, glucose effectiveness. Let's look to see if there's an interaction. And in fact, there is, right? So we were able to show, this is, again, showing you the genotypes for a GCKR, but down here, I took our dietary fructose and just created tertiles so you can see the average sort of amount of dietary fructose. And you can see the effect of fructose on glucose effectiveness. So the higher the fructose, the more glucose uptake there is. Again, we're assuming that that is reflected in liver glucose uptake. And then we can see within each tertile the genetic effect. Okay. So again, this is information that we were able to obtain because we had this unique phenotype to work with. I actually got a second grant after this to actually we based a lot of the liver effects using liver enzyme levels, and I got funded to do MRIs to see if we could replicate this result with using MRI-based liver fat data. And then COVID hit. And you can imagine what happens to a clinical study when COVID hits, right? You can't recruit. So when the restrictions came down, I took all the money that I had left over for doing genotyping and all this other stuff, and I shoved it all into recruitment to try to catch up with my recruitment goals, which we did do, but now I have all this data sitting around, these samples that I now have to get funded for to do the measurements that I originally said I was going to do. But basically this is, again, part of that idea, the interaction with fructose actually also helped us identify other associations. If you look at the literature for PNPLA3, there's very consistent associations with very specific phenotypes. Beyond liver enzymes is one, but there are a few others. But people have never, for example, shown any hint of an association with insulin sensitivity. And we are able to show that based on the interaction with fructose. So if you're interested, go check out the paper. We've done a whole series of different analyses looking at those 3 different loci. So summary, right? So ignoring the glucose-insulin feedback loop can lead to mechanistic misinterpretations. So again, mostly for the younger folks in the crowd, pay attention to that. If you do rely on using the disposition index, make sure you correctly compute it. Again, I would refer you to the studies by Steve Kahn and his group, where they've actually looked at different ways to calculate the disposition index and show which ones work and which ones do not. But again, you also have to correctly interpret it. Okay. And then, detailed phenotyping of the type that we do does have its advantages. I recognize it also has huge disadvantages, like the fact that nobody else does this, so getting replication is very hard, except wherever Carl is. Where's Carl? Way back there. So Carl works with a group at Wake Forest that have the exact same phenotypes, so we get to try to use them to replicate things. And again, I mentioned the fact that training with Mike as a postdoc. Earlier today, this Ming Yao was telling me I was actually Mike's second postdoc. She said Sumitra Ghosh was his first postdoc, and I said no, sorry, Sim doesn't count. Because Sim was really in Francis's group so anyways, technicalities, right? But I left Michigan. It was very hard to leave Michigan, actually. In fact, at that time, I had a job offer from Michigan and I had a job offer from USC. And really, I could have stayed here. The only problem was I'm an only child, my parents were getting older, and I just felt like I had to be closer to my parents. And boy, do I need to be closer to my parents, because my dad and my father now is on that slope for cognitive decline, and my mom's her memory's starting to get a little worse. So anyways, so making that choice was the right one, but I could have easily stayed here. But I went on to form my own group, and these are my trainees, both current and past. And my group and my career, everything I've done would not be what it is if it wasn't for my two primary mentors, Rich Bergman, my PhD mentor, and Mike, my postdoctoral mentor. And I rely on both of them still. And Mike, I'm sorry, even though you're retiring, you're not off the hook. I'll still be calling you for stuff. I've always relied on them. And Mike has created an environment here at Michigan, as Sebastian was describing, that is just absolutely wonderful. And it really did enrich my life and my career, and I'm ever grateful for that. And, to close out, congratulations on retirement. You like my little trick to get rid of Q&A time?